Hey guys, how are you doing? I'm liking seeing that half of you are struggling to actually get started with your revision. Some of you are saying you're kind of going a bit crazy being at home. How are we all doing, mate? We all right? Where are you? There you are. Any idea when you have UCAS marks? Not a clue. Hi. Yeah, I'm really good, thank you. Yeah, I've had a pretty good day so far, although it's getting pretty depressing that it's like raining outside. It's not ideal. I like the last few weeks have been fairly nice. Like at least it's been, well, we've been inside, but at least it's been sunny. But being inside with it like rainy and depressing is a bit rubbish. Don't know. What are you guys saying? Yeah, with the year 13 stuff, like people, why would you be watching this if you're in year 13? To be fair, if your school gives you a bad grade, you don't have a whole lot of choice but to do an exam, right? So I don't think there'd be like I'm not completely surprised that there are year 13s here. So they don't have to sit an exam, but if they get a really bad grade, which I definitely knew a lot of students who like over the few years have probably worked considerably harder in year 13. So me as an example, back when I did my exams, you could resit if you didn't do very well in year 12. And I definitely resat quite a few in year 13. So my year 12 grades probably wouldn't have got me into the university and stuff that I did end up going to. So I understand. I understand. What were we saying though? Can't even do my sanctioned exercise. That is very sad. Yeah, and I think that's also a very good shout. So, how many of you guys are doing something like biology related at university? Because if you are, then that's another good reason to make sure you're still doing work. Alkaline oil. I was waiting for someone to ask that. Yes, so I do focus on AQA, OCR, EdXLA, EdXLB as my main things. However, I am starting to familiarise myself with the Welsh exam board at the moment and the CIE A levels, the international ones. They basically overlap a lot. I'm just not using them as my like absolute go-to thing sort of thing. So I'm making sure that I'm hitting the big four in the UK. But if you're in Namibia, which is amazing, I've always wanted to go to Namibia, then yeah, I'm sure there'll be enough things which are related, but just be careful with your spec and make sure that things are relevant. You find biology really hard. Oh, any tips on revising plant stuff? It just won't stick in your head. So, well, with any revision, I'd say you just need to assess yourself a lot, right? So as soon as you've learned something, do an exam question to make sure that you've actually learned like what you need to learn. Yeah, medicine at uni. If I were doing medicine or if I was hoping to do medicine, I certainly wouldn't stop working now. Ooh, yeah, good point. I'll get to that in a sec. What else are we saying? BMAT revision 2 is also something which people will have to do. Can you watch this as a year 11? You can do. There's YouTube. You can watch whatever you like, but I imagine lots of it will be very hard as a year 11. Nice. Right, yes. So, Gum, the person who just mentioned this Instagram thing, we are doing something. So, not only do I do these sessions, that wouldn't be in my job. That would be, I don't know. I don't think that I'd pay enough for to be my job. I also do like lots of these web classes from my website. So, SnapRevise, who are behind me over there, there you go. We do lots of these web classes every day, and we do lots of different subjects. And yeah, if any of you like what you've seen today, then if any of you basically follow us on Instagram and share a picture of you like enjoying this session, I don't know. This one isn't the best, if I'm totally honest. This is one of our previous winners, I believe. But if you can post something on Instagram of you having a great time during this, I'm going to pick through the creative ones and try and find the best one. So someone can win a free account. And I don't think it's for a whole year, but it will be for a little while. I don't know. You'll get a free account, which is worth about £50. So for any of you who want to do that, make sure you do before the end of this week. And also at the end of this session as well, I'm going to have a coupon code. Okay, cool. I bet most of you could guess how much that coupon code is worth. Yeah. Any other standard question? What are your standard questions? Ooh, before anyone asks, because there's always someone, it'll be an hour before you ask. I know you're going to ask it. And it is related to the specifications I mentioned earlier. Okay, so let's get started then. So we're going to be looking at membranes. So we're starting with the year 12 topic of membranes, and we're basically just going to be going through until, I don't know, until we stop doing the YouTube sessions. I think the plan is to do these pretty much throughout the lockdown in the UK, because I don't know. I know that lots of you probably don't have a great means of studying. I know that like you don't properly learn how to revise and study until you probably get up to your degree or something. So hopefully this can be just a nice little constant throughout the next few months, right? And we'll just keep going. Only one person does win. Yes, you're right. Although I did find out the other day that your odds are something like, you're at least, what was it, like 500, 600,000 times more likely to win this than you are to win the lottery. And they're basically the same thing. So, right, anyway, ignoring that too. So the graph here then shows the rate of movement of 4 different substances across a membrane. So you got your axes with the rate of movement across, and you've got your concentration difference across the membrane too. The substances shown in the graph are carbon dioxide, testosterone, ethanol, and sodium ions. Which of the lines A to D represents a pattern of movement of sodium ions across a membrane? What do you guys think? Oh yeah, Ken did get cancelled, so Georgia wasn't very well. So C's and D's at the moment. And a B. I don't know. F is a good shout. Okay, so why are we saying D? So you're right, it is actually D. Why do you think it's D? Those of you that have said it. Oh, that's quite a good circle. Ken was cancelled yesterday, yeah. Yeah, good. So it's facilitated diffusion, right? So yeah, A is nonsensical because it's never going to start to increase in rate. That's craziness. B and C are basically the same. So you've got to work out if it's either B or C or D, right? Or B/C or D. So equilibrium has been reached. That's not the reason why it is starting to plateau. So it's not to do with equilibrium here. Why is facilitated diffusion? What's the problem? There you go, Sarge, you've said it. So essentially what's going on is if you'd read this question, the things that you need to get into your mind that need to stick out to you is firstly, we are looking at sodium ions. Okay, if it is an ion, it cannot just pass through the membrane. So because it's an ion, it needs to use facilitated diffusion. Okay, which means that it needs to either use a channel or it needs to use a carrier. Now, essentially what happens with facilitated diffusion, as you increase the concentration difference, or as you increase the gradient, then your rate's going to get faster and faster and faster and faster up until the point that in your membrane, which is going to look something like this, in your membrane, every single channel is going to be occupied. So every single channel has things moving through it. So there's lots of little ions going through, sodium is positive. And at this point where your channel is completely saturated, it just can't speed up anymore. Okay, so what are you saying? Carrier proteins, yeah, I would say that it's probably a channel if it's a sodium ion, but it could feasibly be a carrier. But on the whole, I think it's probably a sodium channel as opposed to a carrier. Okay, I'm not too sure. It's a bit of a weird one, really. So for substance, so it's probably carbon dioxide and testosterone being B or C. They're both at the same rate. Yeah, I think it could be sort of either or, really. But because they're both lipid soluble, they can move, they can just diffuse through. So A is just the nonsense one, and D is like the main one. Okay, so an investigation into how a change in sodium chloride concentration affects osmosis in potato cells concluded that the isotonic point of the potato was 0.25 molar. Okay, which of the statements A to D describes what is happening at the isotonic points? Okay, so at the isotonic point, does it mean that there is a net movement of water from a sodium chloride solution into the potato cells? Does it mean that there is a net movement of water from a cytoplasm into the sodium chloride solution? Is there no movement, or is the movement of water from potato cells equal to the movement of water out? Blumeneck, there's a lot of you today, aren't there? How many are you? 88. Nice. Good job, guys. Yes, we are right. So I was thinking that all of you, I don't think all of you, I thought some of you would go for that one, right? I thought a lot of you would say that there's no movement, but what you need to remember is diffusion and osmosis and all of these things, they are always the net movement. So net movement doesn't mean that there's no movement. It means that there's equal movement. Or it doesn't actually mean that. Net movement means like the overall movement. So you sort of take away the movement in one direction from a movement in the other direction. If there is no net movement, or if there's equal movement, that is what happens when you reach the isotonic point. So it is D here. This is why if you have if you wear contact lens, do you have I don't know, you can store them in that solution stuff. I'm basing this solely on my mum. Essentially, the solution will say on the back that it's isotonic. It means it's isotonic to your eyes, so that it doesn't draw water out or put moisture in. Yes, well done. Yeah, so isotonic means that there is no difference in water potential. So there'll be no osmosis going on, right? There'll be movement, or there will be osmosis going on. There'll be no net osmosis in either direction, right? It will just be equal. Okay, so the fluid mosaic model describes plasma membranes of all living organisms. How does the fluid mosaic model describe the structure of a plasma membrane? So hypotonic is where it's low in terms of the number of solutes. So there's lots of water, not a lot of solutes. Hypertonic is where there's lots of solutes compared to water. Okay. So phospholipid bilayers, that is related. Mosaic because each particle, what particles are we talking about within it? Contains many elements, so intrinsic and extrinsic. Mosaic means there are proteins. Yep, good. Fluid because of movement. Jacka, I like what you're saying. Fluid mosaic is dynamic, so it's not staying the same. It is dynamic. It is moving. I like that. Ooh. Okay. Hydrophobic head proteins, interesting. Cool. So hang on, let's just get to the right bit. So yeah, we want to say that the fluid mosaic model, right? We're talking specifically about phospholipid bilayers. So not that you need to say this, but we are talking about phospholipid bilayers, right? So the things where you have mostly your phosphate heads, and I don't know why I've drawn another circle inside of them. That's weird. You have your phosphate heads, but every now and then you have a little sort of protein attached to it, right? You have a little protein. Some of them might go all the way through. So some of them might be intrinsic. So something like a channel would be intrinsic. You have little bits of cholesterol in there as well. But mostly it's phospholipids, right? So the name the name fluid mosaic, or the model, which is defined as being a fluid mosaic, exists because firstly, it is fluid. Okay, so the fluidity relates to the phospholipids and the proteins being able to move. And this is something that I don't think we really consider much as biologists, because we learn things by images, right? But if this membrane were to be like a real representation, then this protein here could move about in this direction. This phospholipid could move about in this direction. Basically, it's not static. It's constantly moving. Right? You can sometimes get these flip from side to side as well. And that's called flip-flopping, which is a great term. But it's not still. It's constantly moving, and it's kind of similar to your cytoplasm. So I think we have this idea, or lots of people have this idea that the cytoplasm is just static. Everything inside moves about, and you have things called cytoplasmic currents, which move things about. But I digress. It's a fluid because everything is capable of moving. So it is dynamic. I like that word that someone said. Everything moves. Someone said everything moves laterally as well. That is a nice way of saying it, because moving to the side. But everything moves. That was enough on this mark. On this question to get a mark. And then in terms of being a mosaic, essentially the reason for that is because it is embedded with proteins. Right, so if you picture what a Roman mosaic would look like, with lots of different pieces, that's basically what this looks like. You've got loads of little proteins. Okay, so what are you saying? They do vary in shape and size. That's good. Yeah, no, I like that. I'll do that. So embedded with proteins of various shapes and sizes. I think that makes it sound even nicer. Can I read the mark scheme? For here, you probably wouldn't discuss cholesterol. Cholesterol just sort of reduces fluidity. So the mark scheme just says membranes consist of a phospholipid bilayer. Proteins are secreted in the membrane, making it like a mosaic. Phospholipids and proteins can move, hence the fluid nature. So it's not floppy. Nice. Cholesterol on the outside layer provides strength. No, cholesterol doesn't provide strength. It just prevents it from being fluid. Why is it rigid but also fluid? It's not rigid. It's pretty fluid. Can I do glycolysis? I'm not going over glycolysis today, Joshua. Look back on previous lessons I've done. There's definitely one on YouTube I've done about glycolysis. Have a look. Okay, so transmembrane proteins are involved in the transport of sugars across the plasma membrane. So glucose can be moved into cells by facilitated diffusion using proteins called GLUT proteins. There are also some called sweet, I believe, or that might be sucrose actually. Anyway, so these proteins expose a single binding site on one side of the membrane. Glucose binds to this site and causes a change in the shape of the protein, so a conformational change. The change moves glucose across the membrane and releases it on the other side. Explain why facilitated diffusion via GLUT proteins requires no metabolic energy. Now, Murata, what you just said was definitely right, by the way. I don't know if you retracted that or if one of us retracted that, but that was good. If it needs to go, right, facilitated diffusion, right, this is a top tip. For those of you that mention lipid soluble, if it's facilitated, it needs help. Right? If it's facilitated, it needs help. This means it is not lipid soluble. So it's probably polar, charged, or big. Okay, so concentration gradients. Yes, very good. So you can get a mark if you say that. So in terms of how this is going to work, essentially, it's going to be moving because it is moving down a concentration gradient. So there is a concentration gradient. But for a second mark, why is it the case that there's going to be this sort of movement around? So how can it actually do this movement? So there is a concentration gradient. So explain why facilitated diffusion via GLUT proteins requires no metabolic energy. Why is it the case that no metabolic energy is needed? It's passive, but something's changed in shape. So there's got to be like something must have some energy, otherwise nothing would be moving, right? Oh, lovely. Asio. Oh, I love that you are a Harry Potter person. So it relies on natural movement of particles due to their kinetic energy. Lovely. So these particles have to be moving. If the particles, this is a really stupid mark as well in my mind. Basically, I'm saying that because I wouldn't have thought of it. But why does it not require metabolic energy? So let's say that I've got this membrane that looks like this, right? Here's a channel. There you go. So let's say I've got a really high concentration of glucose up here. In fact, it was a carrier, wasn't it? Not a channel. It's going to look like a little Pac-Man. There you go. So glucose is going to come along. And the mark that you need to be able to say is that basically this process will rely on the kinetic energy of this glucose molecule, right? So it doesn't need any external source. It doesn't need ATP or anything. It can just rely on the kinetic energy which glucose has. So because glucose is moving, that energy is enough to provide this movement across the membrane. I think that's a bit of a strange mark, but well done for guessing it, person who did. So it relies. I guess you could relate this to Brownian motion, right? If something has kinetic energy, it's going to start to undergo Brownian motion. And if there happens to be a greater concentration on this side, right, and there's something which will facilitate the movement through the membrane, then naturally it is going to start to move across just due to the random nature of Brownian motion. But yes, I think that is a bit of a leap, that answer. Cool. So where does the kinetic energy come from? Kinetic energy is, right, I apologize if any of you are better at physics than me, because I basically haven't done any physics since year 11, apart from teaching it to year 11 stuff. I haven't got an A level in it. But kinetic energy is just the energy associated with movement, right? It's just like a store, or it's not a store, it is a store. It's a store of energy associated with moving objects. right? So essentially, if it's moving, it's got Brownian motion. It's got kinetic energy. Brownian motion is basically a really nice way of sort of observing this, because I'm sure that's what you want to be doing with your Wednesday afternoon. If you have some water in front of you in a little pot, right? If you put some little seeds on top of it, those seeds will not move in any sort of particular order. They will move. They're not going to sit still, but they're going to move around randomly. That might go that way. That might go that way. That might go that way. And it's caused because all the particles that make up the water are bumping into one another and bumping into the seeds. So it's just the random movement. Brownian motion is just random movement, right? And random movement is what ultimately drives diffusion. So random movement on this side, and random movement on this side. If there are more particles in my left hand, they're naturally going to move in that direction, just randomly. Like if it's a closed container, they're going to spread out from high to a low concentration because they're moving randomly. No, Brownian motion is not on the spec. Sorry, no, it's definitely not. Although I don't think it is anyway. Brownian motion is just how this happens. Kinetic energy is something you definitely will need to know about, though. You definitely can't get away with not knowing about that. Okay, so why is it the case that glucose can't simply pass through a cell, through a membrane by simple diffusion? No, not really. Yeah, so it is too large. So glucose, and this is one which is kind of what I've always taught people as being the limit, right? In terms of molecules' ability to pass through a membrane, essentially the biggest, or glucose is just about on the border, right? So it's just about too big to not be able to move through. So glucose just about can't. Something a bit smaller like oxygen, that can move through. Something like carbon dioxide, that can move through, right? Glucose just happens to be a bit too large. Too large. And then in order to get the second mark, in terms of being polar and non-polar, I'm not convinced. Is glucose polar? I don't think I've heard glucose being, it dissolves, doesn't it? So yeah, it is polar. So yeah, feasibly you could say it is not lipid soluble. That could be something you could say. I think that would be a nicer way of saying it. You could say, go on, we could say it's polar then. And basically you want to give the impression that the phospholipid bilayer is only partially permeable. It's selectively permeable, right? So therefore we want to say it's too large, not liposoluble or polar or whatever, which again, I'm not 100% sure. I'm just going with it can dissolve, so therefore it probably is polar. Correct me if I'm wrong. But we want to say that the partially permeable membrane will not allow certain things through. So why can it not move through? It's too large to move through a partially permeable membrane. That is the second mark here as well. So 2 marks there. If it's polar, then charge cancels out. Hang on. Water can't diffuse through. Water actually is polar. Yeah, so water can't diffuse through either. But this is a common misconception, which it used to get addressed a lot more. But there are things called aquaporins even, right? And H2O can go through those channels. It just happens to be that there's loads of them. Yeah, so it dissolves in water. I think that's probably enough to say that it is polar. So yeah, water in itself cannot move through a membrane. Everyone thinks it can, but it can't. It goes through things called aquaporins. Why is something polar? Basically, if it has an uneven distribution of charge, so a little bit of charge imbalances. So because it's got oxygens and hydrogens in glucose, I guess that is enough of a reason why it's going to form hydrogen bonds with water, which would make it polar. So how do you know? Essentially, if there's lots of oxygen, lots of hydrogen, it's probably going to be polar. Some really large molecules, they just tend to be insoluble because they have like certain things around them. So someone asked me today about collagen. Why is collagen insoluble? Apparently, all of the amino acids on the outside of collagen, they just happen to be non-polar. So collagen has this interesting structure where all the outer amino acids are just non-polar. Are aquaporins channel proteins? Yes, they are. They're channels. Niasi, feasibly, yeah. I'll talk to my boss at some point about that. Okay, cool. Right. So yeah, so this was 2 marks up here, and you could have said sort of one of these things. But they wanted you to say this, which I found interesting. So you need to be able to say that the membrane is partially permeable. So maybe that would be how you'd start this sentence. So why can it not move through a cell membrane? Maybe the first sentence would be a membrane is partially permeable, first mark, and therefore certain things such as glucose are too large to move through it. No, glucose isn't partially permeable. Membranes are partially permeable. What's example? I take all of these from an absolute range of examples, so I honestly couldn't tell you which one it was. I go through like 4 and I make sure they are relevant to everyone on the whole. That one was relevant to everyone. So which option, A to D, describes the role of cholesterol in the cell surface membrane in the human body? So yeah, sorry. What is the role of cholesterol? It binds to phospholipid heads, increasing the packing of a membrane, therefore reducing the fluidity. It binds to fatty acid tails, reducing the packing of the membrane, therefore increasing the fluidity. It absorbs ATP, preventing active transport, or it binds to the phospholipid fatty acid tails, increasing the packing of the membrane, therefore reducing the fluidity. Yeah, most of you getting this right. So it is actually D. So whenever you see cholesterol, you'll basically see it sort of attached to the tail bits of the phospholipid, and essentially it just makes the whole membrane more stable. So it's just going to mean that you can sort of package in more things and it becomes more stable. So a common adaptation is if you live somewhere which is very hot, then you have more cholesterol in your membranes so that your membranes become a bit more solid to deal with the heat. Okay, so yeah, it just basically reduces the fluidity. The more cholesterol, the less fluid your membrane is. Okay, describe 2 other roles of membranes in amoeba. I haven't actually given you any. So what is the point of a membrane? What do they do? That is an adaptation, yeah. Why does it bind to the tail and not the head? I don't know. It just does. That's where it binds. Hydrogen bonds? I don't know. What is an amoeba? It's a little single-celled organism. Oh, yes, I'm liking this. So compartmentalization, very good. Cell signaling, very good. To allow things to enter. Controls entry, yeah, I'd give that. For respiration, probably not. Probably not DNA or anything like that. Gas exchange, I don't think I'd give that. Transport, yeah, I'd give that. So exocytosis, endocytosis. Yes, all of them have this. Respiratory surface, I wouldn't give respiratory surface. Isolate enzymes and optimum pH, tally, very good. Cell recognition, definitely. Cholesterol is insoluble. Cool. Right, loads of answers here. And I like that you guys have sort of gone for the standard ones, right? One that you haven't gone for is that membranes have little molecules on them called cell adhesion molecules, or CAMs, and it basically sticks cells together. So membranes are important in basically, I don't know, adhering cells together. That's how you get tissues. That's something you haven't said. Otherwise, controls entry. Very good. They have receptors. You could mention that. I guess lots of you said that in terms of cell signaling, didn't you? So cell signaling is fine. Recognizing self. Although maybe an amoeba wouldn't do that so much. And that's definitely not how you spell self. So maybe not that one for an amoeba. It would definitely be something that does compartmentalization. That is one of the most important biological or like functions of a membrane, right? So if you've got lots of enzymes here, which I don't want spilling into the rest of my cell, you can just stick a membrane around them and it's separated. So compartmentalization just means like separation. Lots of membranes obviously have enzymes on them because they're embedded with proteins. So enzyme reactions is another fairly good answer that you could have said. And that's pretty much it. Both exocytosis and endocytosis are examples of active transport. They both use ATP. So yes. Self-signaling and cell recognition. Signaling is where you like, I don't know, they're sort of linked intrinsically, those two. So self-signaling involves lots of other things too. So the production of cytokines is involved in cell signaling. The recognition of self is basically where you have a little protein on the surface, which say a white blood cell would go around and recognize by binding to it. Yeah, so I would say really important things to mention here. If I saw this question, I would probably go for that one as a really main one. I'd probably go for this one, talking about receptors on the surface, and probably that one. But they're all pretty useful. I think that one's also pretty good too. What do I mean by receptors? Little proteins. Glycoproteins, glycolipids, yeah, exactly. Well, you get some receptors which are just proteins too. So one way in which substances can cross cell membranes is by active transport. Describe the mechanism of active transport. Can I explain the enzyme reaction point? Basically, there are enzymes on the surface of membranes. So for instance, lactase is on the cell surface membrane of the cells of your ileum. So if you want to find lactase, the only place you can find it is on the cell surface membrane, is on the membrane of your epithelial cells in your ileum. Anyway, compartmentalization is, yeah, these things are just like, that one was just a truth, right? So you could say compartmentalization in an cell and you'd be fine. So describe the mechanism of active transport, sorry. Upper concentration gradient, very good. Using ATP. Very good. Anything else we could say? So for 3 marks. Change of shape, very, very good. There is a conformational change of shape. What kind of protein does it use? Carrier or channel? Against concentration, using energy from ATP. I like that we're saying using energy from ATP. I like that we're not just saying using ATP. So using energy released from ATP hydrolysis is a better way of saying it. It is a carrier. Good. So describe the mechanism of active transport. So let's say, so active transport is used to move substances against a concentration gradient. And then instead of saying using ATP or using energy from ATP, I'm going to say using energy released from the hydrolysis. That's the best way of saying that. ATP. So that's your first mark. That's the second mark. We could say this causing a change of shape. It's another mark. In carrier proteins. Okay, something like that. How does active transport cause conformational shape change? Basically, when your molecule goes to bind to your carrier, right, so it will bind to it, energy is released from ATP, and then this causes it to change shape, so that the whole thing maybe rocks around. So the whole thing moves around, and that way you can then release it onto the other side. Minus 36.6 kilojoules of mole release, kilojoules per mole release. Nice. Lovely. Right. Okay. Figure 6.1 shows a diagram of a plasma cell membrane. So outline the functions of the following components of the membrane. So what is K doing? What is K? It is against concentration gradient. Carriers have to change shape and are used in facilitated diffusion and active transport, whereas channel proteins are just used in facilitated diffusion. Channel proteins, yeah, this is asking you what the function is, not what they are. What do channel proteins do? Good. So, okay, I like that. So it allows a movement. In fact, let's say that allows movement of, you could say small, ionic, I don't want to say charged, can't mean the same thing, polar, water soluble molecules, I think. So it allows the movement of small ionic polar water, polar dash water soluble molecules. Right, so they're the ones that move through. On the whole, this area here is filled up with water. Right, that's what most of your channel protein is. So in order to go through it, it needs to either be ionic, so it can dissolve, polar, so it can dissolve, or water soluble, so it can dissolve. Okay, what is L? What is the point of L? What is it like as well, I guess? Can recognize nervous impulses. It's a glycoprotein, yes, good. So this is a protein, as you can tell. These things are the little hexagons. These are sugars. So it's a glycoprotein. What do they do? So it could feasibly be a receptor. Cell communication, cell recognition. Yep, that's on my mark scheme. Cell recognition. Because a glycoprotein is attached to a protein and a glycolipid is attached to a lipid. Nice and easy, that one. So cell recognition, you could say adhesion. So adhesion of molecules together, adhesion of cells together. You could say apparently binding sites. So yeah, receptors is covered by that. Binding or receptors. One of those would be good. Yeah, binding site for large molecules. I'm happy with that. M, what is M? Could you say cell signaling? So they haven't given it on the mark scheme, and I guess that's because cell signaling is quite vague. So feasibly, it could be involved in cell signaling, and like I would very much be under the impression that something, I don't know, I'm thinking of like the role of adrenaline and glucagon, and they bind to proteins on the surface. They don't bind to glycoproteins. So I would say, instead of saying cell signaling, just say cell recognition. I think that's safer. If it were me, I probably wouldn't say cell signaling. I'd say cell recognition. So M, I don't think is actually meant to be. Oh no, yeah. So sorry, I was looking at N there. That was really stupid. M is a phospholipid, as you say, and its role is it is a barrier, right? So it's a barrier, or it is a member, or I guess a partially permeable barrier, or whatever. It's a barrier allowing. What can we say? Allowing movement of lipid soluble substances. Yeah, something like that. So a barrier, and maybe you want to say like only allowing the movement of lipid soluble substances, because that's all that can move through a membrane. So something like that would be good. I don't think you'd necessarily need to say all of that, but if you wanted to be really, really, really pedantic, which you probably do, I'd say that's M is here. Yeah, it's a phospholipid bilayer. Oh, I see what you mean. No, it's meant to be pointing to the whole thing. So yeah, I can see why you think that. No, it's pointing to the whole thing. Yeah. No, apparently in my mark scheme, it's talking about the whole thing. I guess that I don't know. Yeah, no, it wants the whole point of this membrane. Lipid soluble or just hydrophilic? It would be hydrophobic things that can move through, as opposed to hydrophilic things that can move through. So things that don't like water, but it'd have to be able to dissolve in a lipid to move through the membrane. N is indeed cholesterol. What does cholesterol do? Reduces fluidity. Good. Very good. Lovely. Cool. That's what you need to say. Okay. Give two similarities in the movement of substances by diffusion and osmosis. Alex, that's also good. United one. That's probably pretty good, yeah. Nice. Very good. Yeah, you guys got that really quickly. So both down concentration gradients. How nice would this question be in an exam? So both down concentration gradients. Both are passive. I would always say after that, no ATP or no energy from ATP needed or something. I think you'd get it, but again, I think just an anxiety in me in an exam would just be like, I'm going to write everything I physically can, just in case they don't understand what passive means or something. I think you'd be fine, but okay, right, getting a bit harder then. So a scientist measured the rate of uptake of a monoglyceride and a monosaccharide by the epithelial cells of the small intestine of mice. A monoglyceride is a molecule made up of glycerol with one fatty acid. She did this for different concentrations of monoglyceride and monosaccharide. Use your knowledge of transport across membranes to explain the shape of the curve in the graph for the uptake of monosaccharides between concentrations, between A and B and between C and D. So what is going on between A and B and what is going on between C and D here? ATP is not actual energy. ATP is a molecule. When you hydrolyze the bond between one of the phosphate groups, it releases a bit of energy. So use your knowledge about transport across membranes to explain the shape of the curve. So why is it the case that as I increase the concentration between A and B, why is it the case that the rate is increasing? You can get some marks for basically saying what's going on. So yeah, if you literally just say that between A and B, facilitated diffusion is being used, you do actually get a mark for that. So that's a nice, easy way to, if you can work out that a monosaccharide is probably too large to go through, even without looking at the graph, you can get a mark just saying that facilitated diffusion is being used. This has nothing to do with isotonic points at any point. As concentration increases, rate increases due to a steeper concentration gradient. Oh, I think you actually might get a mark for that as well, which is also interesting. How do I know it's how do I know that diffusion's going on here? Or how do I know? What else can we tell? More enzyme substrate complexes. When there's an excess of carrier protein, which gives an increase in rate. Monosaccharides carrier proteins are saturated, so it plateaus. Okay, so someone is getting this mark over here. There's another mark we can get as well. It's been used. Okay, so see, I think this is a bit of a stupid question again here. Oh no, you can actually get another mark for that. So right, facilitated diffusion is being used between A and B. It looks a bit tenuous how they're giving the marks here. So I don't think they can. It looks like you need a description of what's going on. So right, I'll go over the mark for C and D because that one's easy. So why is it the case that it begins to slow down? Why does facilitated diffusion slow down? I think lots of you said this. So all of the channels are saturated at this point, right? So at 3, between C and D, for the third mark, you want to say all channels or carriers, because I don't think we necessarily know. So all channels, carriers are saturated and the rate cannot increase any further. Right, there is another stupid mark, right, which sort of links to A and B, and we sort of want to be saying what's happening. So I guess this is, you've sort of mentioned about how we know it's facilitated diffusion, but how does this graph show us that it's diffusion that's going on? I know this is a bit tenuous. Saturated means that all of the enzymes, because these little proteins are basically enzymes, means every single one of them is filled. So think about it like an active site, right? All the active sites have been filled, so the rate can't increase any further. No, I'm going to tell you, I don't necessarily think I'm going to get it. I don't think I'd get this. So you get a mark for saying that this is diffusion I apologize for this, because basically because the external concentration is proportional to the rate of uptake. So you can get a mark for saying that those two things are proportional between A and B. So between A and B it is proportional, and then between C and D it begins to sort of curve and level off. So I don't like that as a mark. I don't think that's great. I think if it were me, I would be looking for you to say that it's using a carrier or a channel or something. So facilitated diffusion is being used, like, I don't know, using carriers or something. However, they wanted you to say in this mark that we know it's diffusion because rate of uptake is proportional to the concentration. Which is a bit annoying. How's it proportional between A and B? Because, well, it's almost a straight line. I guess it's not quite. It is slightly curved. But as you increase this one is also increasing by roughly the same amount. It's not completely proportional, I know. That makes it even more of a stupid mark in my mind. But literally what I've got written down in front of me on my mark scheme is facilitated diffusion or movement through carrier proteins between A and B. Between A and B, the rate of uptake is proportional to the concentration, which, as we've seen, is not entirely, which is a bit stupid. And 3, all the channels are saturated for the third mark. If there are enough transport proteins. So eventually, because this is like an endless scale, isn't it? Eventually, all the transport proteins will be saturated. Yeah, so I'm not a fan of that. I'm not a fan of that. Anyway, let's look at another question. So the graph is evidence of monoglycerides being lipid-soluble molecules. Suggest how. So how would this, how does this graph suggest that monoglycerides are lipid-soluble? Directly proportional. Yeah, this time it actually is proportional, isn't it? So this time, external concentration is proportional to rate. We could definitely say that. Which means what? There's no plateau. Yeah, that is important. What does that suggest? The rate will theoretically not slow down. Agreed. What does it mean though? If you get this constant like straight up, what does that mean in terms of transport across a membrane? So it's not using carriers and channels, therefore it's using what? Simple diffusion, exactly. So the graph is evidence of monoglycerides being lipid-soluble molecules because essentially, as the external concentration increases, the rate is also increasing. This is alluding to the fact that simple diffusion is occurring where a lipid-soluble molecule moves through the membrane. Right, I'm adding that last bit in, but... All right, one more question. Okay, so the cells of beetroot contain a red pigment called Batalin. A student investigated the effect of temperature on the loss of a red pigment from beetroot. He put discs cut from beetroots into tubes containing water. He maintained each tube at a different temperature. After 25 minutes, he measured the percentage of light passing through the water in each tube. Draw a suitable curve on the graph above. How would you do it? What shaped curve do I want here? What affects the fluidity? Yeah, exactly. what United wants. No, what Namrata said. So temperature does, something like ethanol does, which is a solvent. Yeah, sigmoidal, it's S-shaped, good. So you would want to draw something freehand that looks like that, right? Don't just join up the dots. Don't do the thing that GCSE kids do all the time. You want to draw something freehand. Weirdly, they also accepted that you could draw a straight line here as well, which I'm completely not convinced by, but they allowed that, which is strange. Okay, so the decrease in percentage of light passing through the water between 25 and 60 is caused by the release of the red pigment from the cells of the beetroot. Suggest how the increase in temperature of the water caused the release of red pigment. Breaks down cell membranes. I wouldn't give that. I would say fluidity. So if you said increased fluidity, I would give that. So it becomes more fluid, becomes more of a liquid. If you said it denatures proteins, I would give that as well. Because obviously there are proteins which are going to be in this membrane, and if they're integral and they denature, there's now a big old gap. What else could we say? More kinetic energy, so not brownie moment, Brownian, as in there was a guy who came up with it, it was called Robert Brown. Therefore it is named after him, Brownian motion. Anyway, we'd want to talk about not Brownian motion, but kinetic energy if you were going to talk about that. If you said it dissolves the bilayer, heat won't dissolve the bilayer, so I wouldn't say that. Increases the pigment kinetic energy, so faster diffusion of a pigment. Feasibly, I don't know how big this pigment is. I don't think it would be big enough to leak. Otherwise, if you had a beetroot in your hand, then all the red stuff would drip out of it. If it was like, I don't know. I don't think that would happen. Maybe it would. Yeah, maybe it does. Maybe it is water soluble. That actually does sound quite familiar thinking about it. No, this question is just talking about temperature. So you guys have said this all. So you basically want to say that it will increase fluidity, it will denature proteins. Or you could just say, and really vaguely, this will get you a mark, damages the cell surface membrane. Why did I say denature? Because proteins could denature. Right, guys. So what was I going to say? I was going to mention this. So like a membrane where they separate things, where you have one thing on one side and another thing on another side. Something else that separates things is SnapRevise, because SnapRevise will separate those people who do really, really, really well with those who do all right to maybe quite well. Okay, so in a way, I like to think about SnapRevise as not just being a tool to get the best grades that you feasibly could ever get in your life, and no other hang on, I don't want to get into false advertising here. You wouldn't want to like not get the best grades feasibly possible. So like a partially permeable membrane, which is selectively permeable, we can think about SnapRevise as being the tool that will allow for the diffusion of knowledge to pass through. And ultimately, the more time one were to spend on SnapRevise, actually the rate wouldn't just carry on going up constantly in a proportional way. It would actually show an exponential curve where the longer you spent on, you exponentially learned more and more and more and more and more. I'm glad you guys are enjoying this. Anyway, no simple diffusion. No, this is all, it's active as well in my mind, because you actively have to do things. Yeah, cool. So guys, I will show you a little bit more about SnapRevise in case you have never actually heard about us. If this is the first time you've ever seen SnapRevise, this will be the time, right? So obviously I do these YouTube sessions once a week or twice a week at the moment. However, I do so much more than that. I do web classes literally every single day. So I do web class, well, not on Fridays actually, I started live there. And my rota here, I haven't actually updated for next week, which is a bit dumb, but I will prove it to you by showing you this week. So here is today, the 29th. I did something today, I did something yesterday, something on Tuesday, I did something on Monday. Thursday, Wednesday. I didn't do something on Tuesday that week because I actually had something else to do, which was really horrible, and I won't talk about it. But look, I do things literally every day, and I tend to do more than one session a day. So I do web classes where I go over, I don't know, enzymes, cells, electrophoresis, all that stuff. I do drop-in sessions where I specifically go over things that people want. So someone wanted me to go over transpiration and the heart, so I did that. Someone wanted me to do blood clotting and the immune system, so I did that. Someone wanted meiosis and hemoglobin, I did that. Someone wanted a biology book club, so I made one of them. Yeah, not only do I do these online classes, I also do an absolute ridiculous amount of classes just all the time. So I'm doing about 3 hours of teaching at the moment if you were to sign up. SnapRevise obviously doesn't just offer that though, we also have lots of other very nice things. So for instance, if you wanted to learn about digesting, no, not that. Let's do mass transport in animals, right? Let's say you wanted to do one of these, right? And let's say you watched a video and you were very confused and someone wanted you to, so you had a question to ask, then look, I could reply to you pretty much instantly, which is nice, at just 24 hours, 24 little hours after. But no, our website has basically little quizzes. So as soon as you do start doing mass transport, for instance, there are 14 questions and you can go through them and you'll see what you get right. And by the time you get to the end of a little quiz, and I'm literally just clicking randomly, as you can probably see, it will basically tell you what you do and don't know. So you'll do your quiz. And then if you don't know very much about oxygen, which apparently I don't, it will load up an oxygen video. So you can watch the oxygen video and go through that. And there's a nice man who will talk to you about it and talk to you about oxygen dissociation curves and stuff like that. You can do that. After you've done all the videos and you've spent, how long is that? So just on that topic, that's like at least half an hour. Then really nicely, there is a second quiz. So let's see how we've like done this time. We can see if we've now learned stuff. That was lucky. And if that still wasn't enough, which it might not be, there are some more exam questions. So there'll be 3 more exam questions. And someone will go through the answer to this. So they'll go through the exam question with you. And if you were thinking, oh my God, this still isn't enough, and I thought this was sold to me because it was like diffusion or active transport and I was going to learn at an exponential rate, well, there is also an exam pack. So after you've done that, maybe you'd want to go through some exam questions. And maybe you think, oh, maybe 6 questions would be good. Well, my friends, what about 13 questions? Oh, even more than that. Look, they're not endless. That'd be a lie to say that. But there are a ridiculous amount of questions which you could then basically start to go through. And as soon as you've done the question, bam, the solution is right there as well, just for you to get it. And you can tick off that you've got it, right? And it will basically save what you've done. So it knows what you've got and what you don't got. And then you can come back and learn stuff. On top of that, we also have a nice revision guide too, where there's basically lots of information which you could download and you could stick up on your parents' bedroom or something to upset them. So that is SnapRevise. I know I'm biased, obviously I'm biased, but I genuinely do think it's really, really good. So if you are at all worried that you currently don't have a teacher and you don't want to fall behind and you don't want to get into year 13 and have people being ahead of you and you don't want to get into university and not know everything, then check out our site. You can get like a free account for a little bit as well, just to see what we do, right? You can't really say no, but you can't go wrong with a free account. If you hate it, that's fine. If I haven't sold it to you at all and you think this is rubbish, which is absolutely your opinion, not mine, and it's fine, then feel free to carry on coming to these YouTube sessions and you can set a little reminder if you type in SnapRevise and essentially you can see what we've got. So that's the one I'm doing right this second. Here are some in the near future. And otherwise, I think all I have to show you is here is a discount code for anyone who is excited to join SnapRevise, where if you basically type in this code, you'll get £10 off your first ever like purchase. And there are basically different, I don't know what the term is, packages, that's the one. So there are different packages. So if you want the one with the like live tuition from me, then that's the most expensive one, which is, it makes sense. But it's not ridiculously expensive because I think it works out about £1 per lesson, which is okay. If you want something that isn't quite as good as that one, you can get all the content, like all the videos and stuff, then you can sign up to that, or you can sign up to the cheapest one, which I think is only like £9.99 or something. So we're not going to give you a quid, but you basically won't have to pay much money or any money at all for that. So does anyone have any other questions? I'll stick around and answer your questions for a bit. What are you saying? £50 daylight robbery. Isaac, are you going to uni next year? No worries, guys. Have a really nice rest of your day. You're thinking of studying biology at university, the non-medical aspect. That's what I did. And I was curious about what one can do with a biology degree and the sort of content that is covered. Oh, that is a really good question, Sammy. So I basically did, I chose a lot of varied things. So lots of my friends who did pure biology or zoology have gone on to ecology and things like that. So one of my best friends who I lived with at uni, she went and did a PhD after she finished because she wanted to get into that sort of side of things. And she's now working sort of in that area. I've had other friends who have completely left biology and they've basically just used it as a means to prove that they are clever enough to do another job. So for instance, you could go and work for any big company really, and they'd be pretty happy to have someone with a biology degree because you can clearly do maths and you can clearly do complicated things. Yeah, I think there are loads of things that you'd be able to find. So I never wanted to do the medical things very much. It wasn't my like forte kind of thing. I always preferred the cell stuff and I preferred the genetics things. So yeah, there are things in those fields that you could do. So it depends if you want to go into academia or not. And if you do want to go into academia, you'll find loads of things. If you don't, then yeah, the world is your oyster, really. Like anything in London, like I say London because I live there, anything in a big city, you'd be able to do really, really well. I don't know, teaching. I used to be a teacher. You could be a teacher. That's a good one. Will I be doing more live class on topics? Last year 12 studying at the moment. What do you mean, Jake? What else do you guys said? Have I missed anything? So yeah, the £50 per month is definitely the top package, by the way. That's the most expensive one, and in my mind, it's not real. It's not ridiculous. I know it is a lot of money, and I know that not everyone has £50 to not throw away. But I know people don't have £50 to invest because that's how I would definitely look at it. However, when I used to be a tutor, I charged about £40 per session. So if you're getting, what is it, about 40 sessions or about 45 sessions per week for £50, that's considerably better value than what I used to charge. Because I don't think I rip people off, but you need to get that. What it's a £10 off for? Basically, just when you join our website, you can get like a free account for a bit, but we have different packages, and essentially, like they cost money, obviously. So I think the cheapest package is like £10 a month. I can't remember what the second cheapest one is, but you can get £10 off basically. I'm glad you love these videos, Sammy. That's really good. I'm pleased. Where are your class timings though? As I have morning lessons online with my school teachers. So the times of mine at the moment are 10 till 12 in the morning, and then I do another one at 3 o'clock. And I think chemistry is after me. So chemistry is like 12 till 2 or something. And then maths is like, I think after that, so like 2 till 4. I don't know. Basically, there's like if you Sin Fuja, if you if you basically send an email to support at SnapRevise, I'm pretty sure there'll be a list of when all the sessions are, because I understand that lots of you will probably have sessions going on at the moment from school. What do I do in the book club? So basically, I got my kids who want to be part of a book club to vote on a book. So I had about 5 or 6 books I haven't read, which I thought were really useful to put on a UCAS statement. So we're currently reading one called The Immortal Life of Henrietta Lacks, and we just talk about it. So we read it, talk about it, what we think, opinion, stuff like that. Normal book club stuff. My school's finished these topics, so it's mainly a revision. Will you be doing live streams on topics like genetics, ecosystem? Jake, yes. So my plan is to basically, as soon as I've done all the year 12 stuff, I'll start moving on to the year 13 stuff. If you go back in time a little bit, I know you're looking for live streams. I've done some of them. I haven't done loads of them, but I've done a few of them on like photosynthesis and stuff. But a bit annoyingly for you, basically, I'm going through these for year 12 first on YouTube. Obviously, I'm doing these on my website, but I'm not doing these on YouTube at the moment. Is the offer available for the basic package? I don't actually know, Maria. I doubt we'd give you a quid. So I don't know. Yeah, I really don't know. I'm glad the photosynthesis one was helpful. I'm a nervous system. I'm glad. Yeah, I've done a few. If you have a look around, you should be able to find some that I've done. I don't know. We're doing them a bit sporadically. Like we were doing like the hard topics in YouTube before. Sorry, in A-level biology before this. But as soon as everyone got locked down, it just made sense to us to start doing a few more videos and sort of starting off at the beginning and going that way. Yeah. Whereas I don't know, in the past it was more about preparing year 13 kids for their exams, right? But at the moment, that's not the massive priority, I guess. Yeah, okay. Jake, I think I don't know what I can really do to help you. Like if you were to sign up and check it out, then I could do some work with you. But otherwise, it's going to be difficult. You're going to have to just wait, I think, if you want it on YouTube. Right, guys, I'm going to head off, I reckon. So have a lovely rest of your day. And I will catch you at some point next week on Monday. So have a nice day, guys. I'll catch you later.